I'm your friendly neighborhood microbial ecologist just down the road in Ames, Iowa, at the National Animal Disease Center. And today I'm going to talk about the swine gut microbiota status and outlook. Here on my first slide is an artist's interpretation of one of my new favorite bacteria. It's an uncharacterized microbe that I'm going to talk about in a few slides. So the gut microbiota, just to make sure we're all on the same page. Microbiota simply means bacterial community. So the soil has a microbiota, marine microbiota, skin microbiota, and the subject of today's talk is the gut microbiota. As you may have heard, there are more than 10 times more bacterial cells associated with the human body than there are human cells in your body. And although this figure is often disputed in microbial ecology, it's approximately in the right ballpark, and this is also true for swine. Also, there are over 500 different unique species of bacteria in this gut microbial community. So to give some context to that, think about E. coli, which Fabio just talked about in his previous talk, and all the things you know about E. coli. It's a lab strain, it's a commensal strain, and also some versions of it cause pathogenesis. We know more about E. coli, arguably, than we do about any other microbe, and yet 50% of the genes in E. coli's genome have unknown functions. And so take what we know about E. coli, think about all of these other 500 species, and in essence, we know very little about the vast majority of microbes in the mammalian gut. So how do we measure this vast majority of unknown microbes? So the classic way is to culture these bacteria, which is a terrific way to get intimately associated, to learn intimate information about microbes. However, less than 1% of bacteria are cultivable by what I'm going to say any one laboratory technique. And the reason I say that is because there are some really new and exciting cutting-edge technologies for culturing, including single-cell culturing, and of course just brute force using multiple different culturing methods. And so we can access more cultivable bacteria than we used to, but it's still very labor-intensive and expensive. And so to learn about bacteria, the new fashionable way to study them is using culture-independent approaches. So this morning, many of you may have gone to the session on using genomics and metagenomics to learn about specific bacteria in communities. And also what I'm going to talk about today is the use of 16S rRNA gene sequence analysis. This is an important tool for microbial ecology because we look at one gene. It's a gene that is ubiquitous among all bacteria, so all bacteria have one copy of this gene. And so by sequencing all the copies of this gene in a single environment, we can get a snapshot of what bacteria are present in that environment. We don't learn very much about what they're doing, but we can learn a bit about who's there. So how does this work? So the 16S gene is depicted here. This line, these 1,500 nucleotides of the 16S gene. Green are the so-called conserved regions. You can design primers to these conserved regions, and they sit down, theoretically, on every bacterial sequence in your sample. And then these primers sequence across variable regions. The variable regions are important because these are the signatures of specific microbes. So the variable regions are the parts that tell you that this is E. coli, or this is lactobacillus, or something like that. And so using modern high-throughput sequencing technologies, you can't sequence the full length of the 16S gene. That statement is nearly outdated, but for now it's still fairly true. And so you take primers to a subset of this gene, and you amplify this small piece of the gene. And then what you get on your single tube from your single sample, in our case a single fecal sample from a pig, is you get all of these different copies of the 16S gene in a single tube. Now what do? On the computer, you align all of these different sequences to what's already in the database, and you can put them in piles or bins. And so you can bin all of the yellow ones together because they're similar, all the pink ones, etc. Once you have these bins, you can assign them to who they are based on, again, what we know about bacteria in the database. So hypothetically, these belong to the bacterium called Prevotella, these four are Clostridium, and so forth. And so this row, if you will, is the microbiota of a single sample, in our case of swine gut bacteria. And so this is a really powerful tool because by adding unique tags onto these primers, we can sequence the microbiota of literally hundreds of different samples in a single swoop. Our lab can multiplex, that's the word for doing more than one, we can multiplex over 700 different samples in a single sequencing reaction, and then on the computer sort all these things out. So that's how we do a microbiota analysis. So the human field is far ahead of the ag field in terms of the gut microbiota analyses. And right now it's in a state of correlation. So various states of the microbiota have been associated to health and disease. Notably, states of the gut microbiota have been linked to autism. Microbial production of acylcarotene has been linked to levels of that compound in the drug, which is connected to the brain. It's the gut-brain axis. Also, there's a gut-lung axis showing that, again, states of the microbiota are related to susceptibility to lung infections, such as influenza and indeed asthma. And perhaps most obviously, states of the microbiota are linked to metabolic disorders, such as diabetes and obesity. And so these are really valuable, but some gaps in the microbiota analyses, as you can imagine. Right now, the biggest one is that correlation is not causation. Right now, the field has been in a state of descriptive work, including the work from my lab has been very descriptive, and so we need to move forward to mechanistic studies to bridge this gap. Also, subspecies are often the most important ecological unit in the gut microbiota, and so the analogy for this comes from astronomy, because a snapshot of the microbiota is kind of like a snapshot of the night sky, right? You get a certain amount of information, but really, if you're interested in one particular star in the night sky, much like being interested in one particular microbe in the gut microbiota, you're actually getting very little information about it from this snapshot. And so, a microbial analogy for this really comes again to pick on E. coli. Subspecies of E. coli are quite different. You know, if I detect E. coli in a normal swine gut microbiota, those E. coli's are not pathogenesis factors, right? But at the 10,000 foot level of a microbiota analysis, it's impossible to get subspecies information about whether or not they actually carry pathogenesis factors. And then just to circle back to my image at the beginning, this is my new favorite microbe because it is not yet a defined species. We hope to publish it this fall. This is a scanning electron micrograph over here of a microbe we call BB10, which is related to Enterococcus species. And this is an artist's interpretation of it. We were going for cover art for a journal. We didn't get selected, but it's awfully pretty, right? And so we define this bacterium for its ability to produce butyrate, a volatile fatty acid that's really important for gut health. But because it's not defined as a species, we're not even detecting it in our 10,000 foot level swine gut microbiota analyses. So subspecies are the important ecological unit, but they're often missed in the gut microbiota analyses. Also, membership does not necessarily indicate function. Let's pick on a different microbe here. So the gut microbiota analysis tends to detect microbes at the genus level of classification. And so if you detect Clostridium, of course, many members of the Clostridial genus are beneficial microbes. They also produce butyrate and do other beneficial functions for gut health. But as you know, some members of the Clostridium genus are again associated with pathogenesis. So simply detecting Clostridium in your swine gut microbiota analysis is not terribly informative for what that particular Clostridium is doing in the gut at a given time. And this is a huge can of worms. I hate to even open it. Interactions with the host are complex and poorly defined, right? So I'm going to hide under that one, but my goodness. So think about the complexity of microbiota and then magnify that by the fact that it's not happening in a vacuum. It's happening inside of a host that has genetics, epigenetics, an immune system, is eating things. So, I mean, it's not hopeless, but I suppose I'm starting to sound like it's hopeless. Also, a big question is the fecal microbiota representative? I'm going to show you data in a few minutes that suggests it's not representative of everything that's happening upstream. And then finally, what are the underlying mechanisms of how the microbiota influences health and disease? That's really what everybody's interested in knowing, right? So for several years, 6 years, we've been studying the swine gut microbiota, and we've done a variety of different discrete experiments. Embarrassingly, our largest N has been 12 pigs, which is ridiculous. But we've shown a couple of different things on these 12 pigs per experiment. E. coli populations tend to increase with certain antibiotic treatments. We've shown that certain subpopulations of bacteria, including the Ruminococcaceae, are positively associated with decreased salmonella shedding, so this suggests a putative probiotic target. We've also shown, this is my favorite result, that with Carbiox treatment, Firmicutes populations decrease, but only temporarily. So in the continued presence of Carbiox, we've had Carbiox for 3 weeks, these populations of bacteria seem to recover, which makes no sense to me from a microbiological standpoint. And so we've done these various experiments and shown these interesting things. And then following this, these results, we decided to get a little bigger. We collaborated with a company to conduct a really large trial, 100 pigs, 2 separate years, with an antibiotic alternative, a non-antibiotic feed additive. And so what I'm going to show you here is a preliminary result that launched the rest of my talk. So working with this company, again, we had 100 pigs, 6 months apart, and the important questions of the study really aren't important for what I'm about to show you. But the point is that on the graph I'm about to show you, each dot represents the microbiota from a single pig. So there's nearly 2,000 pigs microbiota is represented on this next plot. So exact same study, 6 months apart. And what you see here, I call this my Pepsi commercial. The red and the blue are completely separate. What we had hoped, of course, is that the red and the blue would be completely overlapping, showing that the microbiotas of these two, as identical as we could make them, experiments, were the same from year to year. And what we got instead was evidence suggesting that the microbiotas in these 2 identical experiments conducted 6 months apart are so different that we can't even lump them together to statistically analyze trends. So this was very disappointing, and we'll still figure out how to publish this, maybe just by looking at one study or something. So we were unable to perform direct comparisons between the years. But what this led us to examine was whether or not we can calibrate our individual analyses of a discrete experiment of the swine gut microbiota. When we started swine gut microbiota analyses, we were among the first groups in the world to be doing it, and now we're grateful that a whole bunch of other people are also doing it. And so we wanted to know what is the variation in a healthy swine gut microbiota. We looked at studies from the literature and their published data sets to address this question. Are certain members of the microbiota always present? Another way to say this is, can we define a core swine gut microbiota to define the health of that ecosystem? And also, does the method of analysis matter? So when I showed you how to conduct a 16S rRNA gene sequence analysis, I showed you, the one region we targeted. Well, there's other regions of the 16S gene you can target. There are different methods of sequencing you could use. Clearly, there's a lot of variability. So does that stuff matter? And so Devin Holman joined our lab group as a postdoc about 6 months ago, right after we got our Pepsi commercial result. And he needed something else to do. And so I posed this question to him, and he gratefully was excited to run with it. So he did his PhD in Canada at McGill University in Martin Chenier's lab. And so he's got a lot of experience with swine gut microbiota analyses. And so he conducted a meta-analysis of all of the available swine gut microbiomes. And so by available, that meant 20 studies, so there's easily double the amount of swine microbiota studies that we were able to access. So a lot of people had not deposited their data in a way that we could access them, and so that was not useful. And also, we required that the data were publicly available prior to March 31st, so he spent about 2 months getting all the data, so that was his cutoff. And then for any microbial ecology enthusiasts, his method was closed reference picking in the program called Chime. We had to use that because, of course, all these different studies had different regions of the 16S gene, and this is the only method that allows you to more or less align them together. So this led us to use over 25 million quality filtered 16S rRNA gene sequences from nearly 1,000 swine gastrointestinal samples. These sequences came from 3 continents and 10 countries. This is a little bit weighted because over half of the data came from studies in the United States. The pigs ranged in age from pre-wean to slaughter. In these data, Devin was able to define a so-called core microbiota. I use this term loosely because in the scheme of things, only 1,000 pig samples is pretty small, and so the core microbiota might not pan out over the years, but for now, over 99% of the fecal samples contained the following bacterial genera. And I found this to be pretty impressive considering the different countries, diets, all the different things that were happening. Over 99% of these fecal samples contained these 5 microbes. Whoops, keep doing that. Sorry. Prevotella. And interestingly, 75% of the samples contained an uncultured member of the Prevotella. Clostridium, Alloprebitella, Pneumococcus, and then a really undefined group of microbes, the RC9 gut group, which has only been described as being detected in swine. The earliest I found was 2015, maybe before that, but I'm not sure. And they have scarce cultured members, so it's related to the Prebitella group, but we really don't know very much about it. And so what I think is very interesting about this is everybody's so interested in the microbiota and manipulating it, and look how little we know about it. You know, of our top 5 microbes that are present in almost all of the fecal samples we looked at across the world, we hardly know anything about any of these. If you know something, come talk to me afterwards. I should say I don't know anything about these bacteria. So there's our core microbiota. So the other thing we're interested in is what factors impact this microbiota status. And so Devin ran some statistics, and the factors he could look at were the GI sampling location. So that included everything from the stomach to feces. Study, and I'll talk more about that in a minute. Country of origin, hypervariable region sequence. Again, that's what part of the 16S gene line that was looked at. The age of pig, and the sequencing platform. So all of these were statistically significant at this p-value. But what's interesting here is this R-value. So the R-value is the correlation of a given variable to the microbiome. And what's fascinating is that the GI location is the strongest variable, with an R-value of 0.5, which in biology is really darn high. And so the gut location, small intestine versus large intestine, is the biggest driver of microbiota differences. That's important because most measures of the microbiota look at feces. And so feces is really not telling you at all what's happening in the small intestine. And then the next thing to highlight is study. So study is a really loaded word, right? Because there's so many different factors that are in that. What's the feed? What's the parity of the sow? All these different things. And my point here is that all of that stuff got watered down in study because nobody, including myself, reported all of those things. And so this is going to be one of my soapboxes for this talk, is that everyone needs to be a better steward of reporting their metadata, because really, unless we record all of this other interesting things, genotype, kinetics, all this stuff, we're never going to figure out what's going on with the gut microbiota. We're already down to 2 minutes, man. Okay, so gut location is the strongest driver of microbiota differences. This is just a plot emphasizing that point, so we can go through this really quickly. Principal components analysis. I know it looks like a hot mess, but basically all of the large intestinal samples group here. The circles are mine, they're not statistical circles or anything. And the small intestine samples group here. So this is just a visual supporting the statistic I previously showed. This is a linear discriminant analysis. So the next thing Devin looked at was what bacteria are statistically overrepresented at a given gut location. And so here you can see the gut locations listed, and these are different genera, they should be in italics, that are enriched at a given location. The 3 things I wanted to highlight here was that, first of all, the colon. I was delighted by this. This is somewhat expected based on what we know about the function of the gut microbiota in the colon, right? This is kind of a gross representation of the food chain here. The Bacteroides breaking down the complex carbohydrates, and then the gram-positive bacteria fermenting those carbohydrates, so this is kind of nice. I didn't care for the fecal result, because I would have liked the fecal result to show that there was nobody different, right? I wanted the fecal result to be representative of the other ones. So I'm not sure what to make of the fact that the feces have signature microbes that are unique from the rest of the small intestine, right? And then the other thing I don't know what to make of is Campylobacter being enriched on the colonic mucosa. So again, if anyone has information about that, I'd love to visit. So to summarize, researchers need to publicly release their microbiome data. That's soapbox number 1. Again, over half of the studies in the literature we were unable to analyze in our meta-analysis because Devin simply could not access the data, which means they either weren't deposited or there were errors in the deposition or something. Researchers need to be stewards of their metadata, deposit that metadata, keep track of it, and deposit it with the data. Gut location is the biggest driver of microbiota differences, bigger than the study effect. And tentatively, we were able to define a core microbiome for now based on what's in the literature, and it included these 5 members. So, markers of swine gut microbiota health. Health is very difficult to define. I'm not confident that analyses of the swine gut microbiota can lead to the definition. I'm not sure that there are biomarkers of a swine microbiota health. What I would guess could be coming is that we get to define a range of expected swine gut microbiota health, much like you can for total blood counts, right? You see white blood cells fall within this range. Well, maybe there'll be a microbiota endpoint, if it falls within this range, then you have a microbiota health or something. And then the reason this is important is because what everyone wants to do is manipulate the microbiota toward health and away from disease. And so to bring back our astronomy analogy, can we take a snapshot of the microbiota using the 16S, much like you take a snapshot of the night sky, and can we actually get to a point where we can harness one particular member of this gamish of information and use that to transform something? And I'm not sure that we can do that right now. So an outlook for swine gut microbiota analyses. The field is progressing from observations to hypothesis testing. Everyone, including in human research, everyone knows this is where it needs to go, and everyone's working to take it in that direction. It's important to conduct network analyses with host factors. So I think one of the things in our group we're working towards is having a microbiologist work with an immunologist, work with a nutritionist, right? Bring integration in the group so that we can not look at the microbiota in isolation, but look at it as it relates to all these other things. And then really, we need mechanistic studies. So host microbe interactions are critical. How to manipulate the microbiota is an area in critical need. And finally, bacterial function. So our graduate student, Julian Traxel, is interested in butyrate-producing bacteria. And so this is some of the ways we personally are actively working towards bacterial functions. Because membership only means as much, membership is very little compared to the functions.